So I'm going to really focus on the center of that business canvas and really think about the value proposition. And hopefully in the next 20 minutes or so, I'm going to tell you why you really need a target product profile, or P for short. And ultimately, the reason behind this is to make sure that you actually develop the right thing. Really something that people will care about. And those people, to Brett's point, patients. They could be the people you ultimately want to. They could be the healthcare medicine to you as a patient. But ultimately, as pointing out, somebody needs to make this. You care about the payers. Ultimately, they are your customers. It's going to be the payers that decide whether they actually you can have access to that product. Really, as Brett was just mentioning, at the end of the day, somebody needs to make money from your science. If nobody's going to make money, it's very, very unlikely that your product is going to move. You have some wealthy sugar daddy, some philanthropist that's going to pay for your research and move your projects ahead. Ultimately, kind of the payers are interested in your product for two things, because their job is to really try to reduce the cumulative cost of healthcare. And they will price your product according to a number of variables. Some products are very, very expensive. You may well have heard of the CAR T therapies. They have a price tag of somewhere to the tune of $500,000 a year. But they are provided to patients because they are actually curative. They will actually kind of give you many years of survival, so that $500,000 a year is worth it. On the other hand, if you're just providing an add-on, a supportive care product, that may actually cost to the tune of $10,000, a lot less, because it's really just an add-on to other medicines or other interventions that you already getting. So clearly there is a pricing differential depending on the products you provide. And the other thing to think about your product as well is barriers to entry. This is important, because if there are accessibility issues, if there are market forces that stop you actually getting your product out there, you're not going to be able to charge those type of amounts. You're not going to be able to sell much product. When we spoke a month or so ago, I showed this slide, and I think this is kind of one I would like to echo on yet again. A quote from George Merck here: "We never try to forget that medicines is for the people and profits follow." Here that's actually going to really benefit patients here. Somebody will pay for it and you will make money from that. So if you have some transformative medicine, you're probably going to make money out of that. So with that said, you really need to think about what is that unmet medical need. What is the healthcare problem you are trying to fix? And it's not just what you're trying to fix. There are certain agencies out there that really regulate access to medicines. And those may be the Food and Drug Administration, the European Medicines Agency. They are ultimately the gatekeepers that are going to prevent or allow your medicine to kind of get into use. And so you're really targeting, as one of your customers, them, because they are going to be making the decisions, are your medicines going to move forward? They will ultimately give you a label. And if you open up any medicine you get from a pharmacy, you open it up and there's always that white sheet of paper inside, and you just throw that straight in the bin. But if you go in and read that, right on the very first page, there will probably be a box like this one here, indications. So isinterest, a human immune deficiency virus integrase strain transfer inhibitor. And every single medicine out there will have one of these here. Soluja, a PARP inhibitor, indicated for the treatment of ovarian, fallopian tube, or peritoneal cancers here. Januvia, indicated for diabetes here, or kind of a hypertension medicine here. All of these are out there, and that is what the agency really cares about. So you need to think about your product is, what is that label going to be on your future medicine? So with that, you need to really start thinking about what is the clinical readout that's going to go into making up of that label, because that clinical readout really then becomes the endpoint of your clinical trial. What are you going to be measuring in your phase 3 clinical trial here that you're going to go back to the agency and say, I tested X hundred patients and I saw this measure drop there. In cancer, it could be survival, overall survival, or it could be progression-free survival. Progression-free survival is a surrogate endpoint. What you really care about is the overall survival, but in certain cancer indications, if you extend survival for 18 months or for 36 months, the agency may approve your trial for that. Typically, at some point during your drug development process, there will be a negotiation with the agency. You will have a meeting. That could be a pre-IND meeting. That could be an end of phase 1 meeting. Or typically before you start your registration trial, you will have a dialogue with the agency and start negotiating what they want to see. If you do this clinical trial and you have this outcome, will you approve me? And so again, going back to these labels there, as you get in probably into the 10th page, the 12th page, there will certainly be these clinical trial results. And I'm not sure if you can see it sitting there back at the room, but in the case here of Ecentress, it was really a drop in viral load here and viral titers. This is clearly an endpoint of the trial. If you drop the virus load, there's going to be a benefit to the patient's health. Here, for the PARP inhibitor, it was actually an improvement in progression-free survival. For Januvia, it was a drop of a surrogate endpoint. It was glycemic control. And here, in the case of the hypotension agent, there was a couple of endpoints, one of which includes death here. So really, you need to think about what that clinical trial is going to be. And so with that, you really need to think about that's where you're trying to get. So when you start developing your product to actually be able to do that clinical trial, you've got to make sure that your product has actually got the legs to get there. You're going to invest several years, probably 5, 6, 7 years to get your product into those pivotal clinical trials. You want to make sure when you run that clinical trial, you've actually got the right product there, and it can do everything you want it can do. So with that said, you need to really, really aim high. You need to set these target product profiles high for 2 particular reasons. The first is something will go wrong along the way. Your product will look really, really rosy on the first day you make it, and along the way things will start to go wrong. It will start to unravel. So if you aim high and it does unravel, it may still be good enough for purpose. The second thing is you really don't want to let somebody else eat your lunch. You don't want to open up a market. You don't want to show path to the marketplace only for somebody to come along and follow you. You don't want to be the first and then somebody comes along with something better and then takes all your market away a couple of years later. So aim high and hopefully you will be both first and also best in class. So also mentioned kind of complications and barriers to entry along the way. So the other thing you need to think a lot about is kind of for your particular indication or your particular targeted patient population, is there anything you really need to think about? That could be anything from tolerance to side effects. Are there disease complications, and you need to be thinking about those, and kind of that needs to be baked into the product you're going to be developing. You also need to be thinking about kind of, are you going to be combining your new drug with something else? So is there a need to really think about drug-drug interactions here? Is your product going to be used in the ER? If you're in the ER, it's probably good to be able to give something that could be administered quickly. Typically an intravenous is good. If you're going to take your medicine home and just take it multiple times a week, having an oral medication is probably good. Also think about kind of who are you going to be using in a pediatric population? Are you going to be using it in a geriatric population? Both of those populations are very sensitive to kind of the size of pills and the number of pills you actually treat. And also think about if there is any specific market forces out there. Certain indications, Brett was talking about agriculture again. In that case, cost of goods is very, very important. And also thinking about do you need to be using together with a diagnostic? Certain medications out there are used with a diagnostic for patient stratification. The cost of goods typically includes both the drug and the diagnostic, and so you need to be able to price out both of those. So this slide is probably actually quite good. So now you've got your blueprint here, and kind of coming back to the NASA analogy here. So you've got a good idea about kind of who you want to treat. You've probably got a good idea how long you need to run that ultimate clinical study. You've got a good idea about your primary and your secondary endpoints. You probably need to think a little bit about kind of how you're going to be comparing your product to what else is out there in the marketplace. Not just what is standard of care in the marketplace today, but also what may be standard of care in 4 or 5 years' time. You're not developing your product in a vacuum. Other people are competing against you. Other people are trying to get into the same area there. So really think about kind of what that comparator could be in the future. And really think about other complicating factors. So one thing is kind of where you're actually taking your product. So the first part of a target product profile is what's on this slide here, thinking about kind of where you're going. So the second piece of a target product is also now thinking about what you actually need to design. So what is actually the solution you are going to provide out there? And again, this next slide here, you will have seen when we presented it a month or so ago, is a whole concept of a target product profile, in this particular case, for a small molecule medicine. Typically, with a small molecule medicine, you'll be thinking about a number of checks and balances as you go forward. Your first experiments you'll be doing in vitro, testing your product against a recombinant protein. So we really think about biochemical potency here. The second piece, and we'll come back to these and we'll talk through all of these probably slide by slide in a minute, is really about kind of can you modulate your target in a cell? So kind of having a cell target engagement, really showing that you can modulate your target inside a whole cell, and once you've got it through a cell membrane. You'll typically have some form of phenotype, something that's kind of your cells do here that's related to the disease you're trying to treat. And there's probably some form of sensitivity here. You want to show certain responder cells do something, and it doesn't have an impact, negative impact on other normal cells. The next piece will really be thinking about selectivity. Your target is one of probably about 20,000 other proteins in the human body. You really want to be developing something that's selective, only does what you want it to do, and it doesn't have any side effects along the way. Side effects typically mean toxicity, and obviously you probably don't want toxicity with your product. You're going to be using your product in patients here, so thinking about pharmacokinetics is very important. Is your molecule going to be able to kind of circulate in the human body? Is it going to be able to modulate its target in the human body for as long as is necessary to demonstrate a response? And then we're getting really to the most important things here about efficacy. This is really about does it have the outcome in preclinical models, and is that outcome going to be relevant to the human indication here? And then we start thinking about safety. Is it going to be well tolerated at efficacious doses? And so this slide here is really the second piece of a target product profile. This is your kind of your blueprint for what you're trying to make here, and this will evolve as you go forward. So to be able to kind of fill this out, there are really a lot of things you need to think about as you're going through here. So the first is really thinking about having a screening funnel here, and being able to test your products or your prototypes in vitro in a lab. And typically there are 3 components to this, as we just mentioned. Some form of biochemical assay where you're just testing on a recombinant protein, a similar assay in a cellular environment showing that kind of in the presence of serum, getting across cell membranes, you can still do the same thing. And the third point here is really a phenotypic assay. This is your first milestone along the way here to show that you're actually moving in the right direction. Is your solution here going to actually have some disease modifying impact, at least in tissue culture? Often we implement a number of other checks and balances along the way, so some form of biophysical method to show you're actually physically binding to your protein. Typically we try to configure our assays in such a way to make sure that they're not sensitive to false positives. So typically we look for assays, as I mentioned before, that have an up signal. And ultimately we hope that this second and this third assay here are going to actually correlate. And so that you have some, this is now a plot of kind of 300 or so prototype PARP inhibitors that were made for the PARP inhibitor program I worked on in the past. And along the x-axis, you had a cell-based enzyme assay, and up the y-axis here, you actually had death of BRCA mutant cells. And you can see there was a very, very good correlation of all of these different chemotypes, giving us some indication that kind of that our molecules are working on target. And this little red dot in here is actually kind of what is now Zoluja. So I mentioned about proteins, and PARP is one member of an 18-membered family here. And so, there we go. Think about those other family members here and what we're trying to do for them. Do we want them along for the ride? Are those families actually going to be able to contribute to efficacy? In the case of a PARP1 program, both PARP1 and PARP2 are involved in DNA repair. We knew that if we inhibited those, they might actually contribute to efficacy. So in that case, our target product profile, we wanted them in here. There are cases out there where those family members could actually cause us trouble here. We think about multi-tyrosine kinase inhibitors there. Quite often you would have molecules like sorafenib that does work in hepatocellular carcinoma, but it does also have several side effects here. So probably that's no surprise that more selective kinase inhibitors have come along to essentially eat sorafenib's lunch. So beside those other family members that are probably the tip of the iceberg here, there are the other 19,900 proteins down here that low below the surface. And those are the ones here that could quite often give rise to side effects. There are some good articles out there that really talk about the relevance of off-target screening, and there are certain service providers out there that will actually kind of be able to screen for you and provide a safety pharmacology screen of around 100 of these proteins that will help you de-risk your target. So at this stage, we've really got a molecule that's showing activity in vitro, showing a phenotype in cells, but does it work in a whole organism? That's the next barrier along the way here. And you've seen this slide before in the past when I've presented, if you've been present. There are a variety of disease models out there, increasing complexity in vitro to increasing complexity in vivo. And all of these serve a purpose in one way or the other. None of these models, as you can see at the bottom, is perfect, but none of these is imperfect at all. In the middle of the slide, models here are kind of relatively easy to execute. The ones in this particular area are kind of a little bit more complex to operate, but are a lot more disease relevant. So it's important for you to kind of, when you start thinking about a model, think about what that clinical trial is going to be and try and pick a model that's going to favorably replicate what you're trying to do in the clinic. So this actually may be more than one model. You may have some form of workhorse model. One of the ones in the middle is really a good model for testing your prototypes. But then you may have additional models here that kind of really help you to understand what your human PK targets are going to be. Sometimes this actually takes some pretty sophisticated models. So again, this is out of a Science article from back in the early 2000s. And in this case, we were actually testing these prototype HIV integrase inhibitors in rhesus macaque models. So in the US, there are very few macaques out there that actually have monkey HIV, and so we were very, very much limited to how many models that are out there. And so each of these plots is a monkey that has SIV, and you can see where the solid line is across each of these images. That's when we actually dosed 812 here, the prototype integrase inhibitor, and you can really see a drop in viral load, and this really gave us a good idea that this program was moving forward, and this is the seeds of what became raltegravir. There are other models out there, and this is a case of coming back to the PARP story with a BRCA particular mutant tumor here. We were able to demonstrate here as we dose this, we were actually able to see shrinkage. And again, this is the equivalent of a BRCA mutant breast cancer model. So again, gave us some idea this is where we wanted to go. So all of this is good. It starts to give you an idea that you're on the right path, but then you really need to start to understand what are we actually trying to do there in the clinic. And again, you will have seen this slide that I like to belabor, really the thought of a pharmacological audit trail. So kind of, does our drug here in pillar 1 get to the site of action of where you want to have activity? Does it modulate its target? Does it demonstrate some pharmacology? And then ultimately, does it have its phenotype? All of these are fundamental pieces of your program. So in the case of HIV, does the drug get to the liver, which is really the repository there? Is it able to engage integrase? Does it change strand processing here? And then does it drop viral titers? All of these things are important. So coming back to the PART program here, so again, we're looking at 50 mg per kg at the compound concentrations. We were looking at kind of whether it inhibits PARP activity, looking at kind of poly-ADP ribose polymerase chains here. Does it cause DNA damage? Sorry, does it do that both in tumor and in PBMCs? Does it cause DNA damage in tumors? And as we've just seen, does it actually have efficacy? And in an ideal world, you want to be able to do this in multiple models. I just showed you there the BRCA breast model, and there are several other models here that we used. And ultimately, each of these models has led to a different clinical trial. And in an ideal world, you want to be able to do this in independent labs. It's important to do that because that also demonstrates to your future investors or people that are going to later acquire your product that it's actually multiple people can replicate your data. So again, really think about kind of, can multiple people do the same experiment as you? Can they do it in different labs with different reagents? So the other piece that's important here is really to try to think about PK profiles. The PK in a rodent or in a monkey will be very different from what you want to do there in patients. I'm not going to read through this slide here. You can come back and refer to this in the slides that we shared on the web. But we're really thinking about here, what is the PK profile going to look like? There's probably some form of target concentration here that you want to target and then during this time period from 1 to 14 hours here, that your medicine may actually be working. There's no right or wrong profile. A sleeping aid may have a very, very sharp profile like this with a rapid washout. A migraine treatment may come up very, very quickly and then have a sustained plasma concentration. And then others you may even want to think about kind of a once a week dosing, for example, or in the case of an antibody. So it's important to really start thinking about kind of what is the characteristics of that PK profile that you're going to want to do, and that will help you make sure that you're going to be able to have a right product to test in patients. And we typically do that by kind of conducting a series of pharmacological experiments. So again, this is our OXPHOS inhibitor that's currently in the clinic. We went in and we tested it, as you can see here. QD means once a day here. We tested at various doses here with once a day administration. Some of these, if we administered too much once a day, were very, very toxic. You can see these bars here don't progress. But at 5 or 10 milligrams per kilogram once a day, we were seeing some pretty striking efficacy. We also did exactly the same experiment where we administered the compound twice a week. And again, you can see several of these did not show any activity at all. But either what's this, in red at 10 and in blue here at 100 mg per kg, we saw some pretty compelling efficacy. And when we came back and we actually measured the plasma concentrations at trough, so either at 24 hours here, you can see it either 20 or 40 nanomolar here at trough, we saw some activity, or here again in at 10 milligrams per kilogram twice a week, at 72 hours here, there was about 10 nanomolar here, was efficacious. So that really gave us our PK target. So with that PK target, we were able to start to make some predictions of what dose we need to be using in the clinic, and we were able to kind of start to extrapolate. This is important because we really need to understand how much drug do we need to administer to humans. It's one thing here to be kind of giving a 10 milligram dose every day. Can you imagine what it's like to start giving kind of 2 grams 3 times a day? How many of you want to take 30 ibuprofen pills a day? I mean, it's just not going to happen. So in that case, you really want to go back to the drawing board and redesign your product. And there are many ways of really thinking about kind of predicting what that human PK is going to be like. We use a technique called allometric scaling, and that is essentially scaling the PK based on body weight of the animals. We use in vitro, in vivo correlations where we can kind of use liver microsomes or hepatocytes, and then knowing kind of the amount of size of a liver, make some correlation there. So all of those help us to gauge what the human PK is going to be like. And are you at the 10 mgs or are you at the 2 grams 3 times a day? Where are you on the scale there? So coming back to this slide here, so there are a couple of things really to think about here. In vitro, what is that relevant disease phenotype that you want to be measuring? What is that selectivity profile? Do you want to be selective within a protein family or not? What level of kind of selectivity against anti-targets can you have? If you're very, very potent up here, you can be a lot more lax down here. If your target product up here is quite high and you're in the micromolar range, you need to be very, very stringent here because that therapeutic index is going to collapse. Coming down here to PK, really, what do you want your PK to look like? Are you looking for something like a migraine pill where you want it to work very, very quickly? Or do you want something here that's got a much flatter PK profile? Coming back to PD and efficacy, really thinking about which model you want to be used. What does compelling activity look like? I mean, is that completely obliteration of viral titers? Or is that or can you be a little bit more lax there? And again, thinking about all those checks and balances on the pharmacological audit trail. And ultimately think about tolerability. How large does that therapeutic window need to be? Oncology, you can be a little bit more forgiving, but if you're going out there with kind of a diabetes medicine now in 2019, you've got to have a pretty high safety barrier here. So as Brett was also saying, your product is going to evolve during the course of your development. Your target product profile here will evolve, and that is really okay. Your science will help shape what that target product profile comes. And as you go out and you talk to more key opinion leaders, to more stakeholders, they're going to help you to refine that. Along those lines, really think about pressure testing this target product profile on both your future customers, future investors, but also kind of key opinion leaders who may actually administer your medicine in the future. And again, you've seen this slide in the past. Development of medicines really is a funnel. It's going to take you a lot of prototypes before you may actually get one product across the finishing line. And so really get it right the first time you make it. It's going to take you many years to actually get your product into the clinic and then many more years in the clinical development. So it's really worthwhile to take time to optimize your therapeutic. And really, you've got to know where you're going, because if not, you may end up someplace else.